All right, everyone, we're going to get started. Thanks, everyone, for being here. This is our afternoon seminar, and my name is Cinnamon, and I'll be one of your hosts for today. For folks that might be new to us, I'm looking around, you all know this already, but this is a hybrid event, so we have folks online and folks in the room. For folks online, your cameras, mics, and screen shares have been disabled, but please communicate with us through the chat function in your Zoom. Our volunteer, Roseanne, will read out any questions or help you address any technical issues. So feel free to interact with us that way. For folks in the room, you all know this, but when we get to the question and answer, if you just raise your hand, I'll bring you a mic, and that way folks online can hear our questions as well. But again, thank you so much for being here. Wanted to make a little plug for some events that are coming up. So next Wednesday, we have our next mentor seminar in the library. So if you are a mentor of any of our summer interns, feel free to join I Chung in that mentor seminar. Our normal weekly seminar next week is on May 29th. We have Riley Boyd, who is the founder of Disabilities Within Ocean Sciences, who is a graduate student with the Big Fish Lab, is going to be talking about breaking barriers below the surface, navigating challenges and solutions for accessibility in marine science. So I hope everyone can come and join and have a conversation with us about those issues. It would be really great to have Riley well supported, so come on out. I also wanted to let you know that we have next week a lunch and learn on Friday. We have Martin Thiel, who is Managing Director of Marine GEO with the Smithsonian Environmental Research Center. And he is going to be here talking to us about monitoring coastal habitats with professional scientists and volunteers. And in this time when the world is falling apart, thinking about how we can use volunteers to help us with our research, I think is a really important conversation. So that will be Friday in the Gwynn Library seminar room at noon. So hopefully you can join us. But the reason that you're all here is to learn a little bit more about what's going on with our speaker. And this speaker was invited by Dawn Barlow, who is a postdoctoral researcher with MMI. So I'm going to hand the mic off to Dawn to introduce our speaker.
All right, it's my pleasure to get to introduce Dr. Jessica Bolin as this week's seminar speaker. Jessie is a quantitative marine ecologist and data scientist interested in applied research for a more proactive, sustainable, and climate resilient world. And her research interests span seafood sustainability, climate change, risk adaptation, and ecological forecasting. Jessie received her PhD in 2024 from University of the Sunshine Coast and CSIRO Environment in Australia, focused on fisheries forecasting and climate adaptation. And she's now a postdoc at University of California, Davis, focused on climate resilience of abalone. And you'll hear more about all of that today. Jessie and I have crossed paths in various capacities over the last several years because of a mutual interest in species distribution modeling and ecological forecasting, and I've been always really impressed by her ability to bring together a lot of different diverse and interesting data sets and her engaging ability to share her findings and do research that's both scientifically interesting and impactful and applied. So it's a real joy to get to have Jessie here with us for a couple of days. And if any of you want to continue conversations after this hour-long seminar, we'll migrate to Beer One later, and everyone is welcome to join. So with that, the floor is yours, Jessie. Take it away.
All right, thank you, Dawn, and thank you everyone for letting me speak today. I'm really keen to be here. So today I'm talking about fisheries adaptation to climate change, focusing on 2 case studies on broadbill swordfish and red abalone. So first, I thought I'd just share, I'm from the Sunshine Coast in Australia. If you're not familiar with the Sunshine Coast, it is famous for its white sandy beaches, for being the home of Steve Irwin's Australia Zoo, and home of the big pineapple. And like Dawn just said, I did my undergrad, honours and PhD all at the University of the Sunshine Coast in Kylie Scales's lab in quantitative fishery science. During my PhD, I was also a chapter scientist for the Oceans and Coastal Ecosystems chapter of the IPCC 6 Assessment Report. And now I am 1 year into my postdoc at University of California, Davis. So yeah, I'm a quantitative marine ecologist. The things that make me tick are really applied research focused on things like seafood sustainability, fisheries, climate change, marine disease forecasting, and dynamic risk adaptation. So The first case study I'm going to talk about today is the one on broadbill swordfish that comprised my PhD work at USC and the CSIRO. And this is on forecasting and projecting swordfish quality for dynamic industry adaptation. So, one of Australia's largest high-value export-driven fisheries is the Eastern tuna and billfish fishery, which stretches right along the east coast of Australia from the tip of Cape York down to Tasmania. And during my PhD, we work with two of the main longline tuna and billfish companies operating in this fishery, which were Four Seas and Walker Seafoods Australia. And these, like I just said, are the main swordfish companies operating in this fishery, operating out of Mooloolaba on the Sunshine Coast, which is historically a major swordfish port for Australia. And so as such, our industry partners account for the vast majority of swordfish harvested from this fishery. And most of the swordfish gets exported fresh via air freight to high-end sushi restaurants and consumers in Japan and the United States, specifically San Francisco and Boston. So obviously, meat quality is super paramount, right, for our industry partners, because the quality of the fish directly influences the price of that final product. But in recent years, they've been experiencing increasing instances of this jellymeat condition in their swordfish. So the proper word for it is myoliquefaction, and it's where the meat of the swordfish progressively turns into this gooey, mushy-like texture post-mortem. And apparently, this has been occurring way more often in summer or when the waters fished are warmer than normal. And even though this has been purely anecdotal, this has raised some concerns as to the effects of climate change and whether that will exacerbate, the frequency and intensity of these future jellymeat events. And this is especially important for the east coast of Australia, because the main oceanographic feature that we're dealing with here is the East Australian Current, which is a warm, southwards flowing western boundary current. It's a hotspot of marine climate change. We know that it's warming, and we've also seen increasing frequency of marine heat waves in the region. So, jellymeat is obviously a bit of a problem, right? It's disgusting and it can't be sold to consumers. So it would be great if we could find a way to reduce the risk of fishers harvesting swordfish that turns to jellymeat in the first place. And this provides the context for our study on forecasting and projecting the quality of broadbill swordfish. But step one is, how do we even forecast fish quality in the first place? Well, marine ecosystem forecasting is an emerging data-driven discipline, and it seeks to provide information on future ocean conditions to end users in industry and management on a range of timescales, from near real time out to decadal. And the whole point of being able to forecast something is to facilitate proactive and adaptive planning, right? By minimising risk in unfavourable conditions and maximising benefits and opportunities in favourable conditions. And so you can see here, this is the first wave of forecasting tools that have been developed for fisheries in Australia thus far. And they have almost exclusively focused on fish distributions, where they take the habitat preference of a known fish of interest and they forecast how and where those preferred conditions will move on some future timescale. But a stretch goal for these distributional forecasts is to not just predict where a fish might be, but also predict the quality or the condition a fish will be in. And forecasting fish quality could be especially useful for high-value fisheries, where the quality of the fish directly influences the price of the product. So we published this paper, and it reviews the drivers of fish and shellfish condition with an explicit focus on high-value fisheries. We look at environmental variation, parasitic infection and disease, and the interaction between the two, all within the context of marine climate change. We then propose next steps forward and what's required for how we can incorporate aspects of fish quality into the next generation of these fish distribution forecasts. But we realised there was this huge gap preventing these sorts of fish quality forecasts from having already been developed, which was a lack of biological data of fish quality being systematically collected in the first place. And so this gap is what motivated us, again, to do this PhD work on forecasting and projecting swordfish quality off Eastern Australia. So going back to jellymeat, we now want to know what actually causes this jellymeat condition in the first place. So we go to our industry partners and we sampled swordfish directly as they were being unloaded from the boat. We then took those swordfish muscle tissue samples to the lab and we ran a bunch of different laboratory techniques to try and get to the bottom of what this causal agent of jellymeat is. And that resulted in the causal agent being a microscopic cnidarian parasite. This is a mixozoan and it's called Kudoa myoliquefaciens. So the way it works is that it gets into the muscle tissue. It's like microscopic, it's the size of a red blood cell, and it waits for the fish to die. When the fish dies, it activates and it secretes an enzyme into the muscle tissue. And over the course of 2 to 72 hours post-mortem, that enzyme slowly liquefies a tissue and can turn it into that jelly meat-like texture. Now, just as a fun side project, we were also given this loin of yellowfin tuna that had a bunch of white cysts, like tiny little white blobs all throughout the muscle tissue. And they wanted to know what was causing it. And they were also saying that they were getting this condition more often in warmer waters as well. So we did the same laboratory techniques and we found that is also caused by a related species of Kudoa called Kudoa thani. So I found that really interesting that you've got two different species of the same genus of parasite manifesting in these two fish in completely different ways, yet still, completely destroying the quality of the fish. So that's great. We now know what's causing jellymeat. We now want to know the environmental conditions associated with infected swordfish. And so we put this out in Fisheries Oceanography earlier last year. So I want you to look at that map on the left. That is total or cumulative swordfish catch in Australia over the past 2-ish decades. And so you can see that most of the swordfish caught within this fishery was caught directly off of Mooloolaba on the Sunshine Coast. So that map on the right shows sampling effort of swordfish during my PhD. And so we sampled swordfish from those general areas, and that was completely dependent on where and when our industry partners were fishing, COVID dependent. So in total, we sampled nearly 1,700 swordfish over 2.5 years, and nearly 80% of the swordfish were infected with this parasite. And at least half of the swordfish caught during any one fishing trip were infected with this parasite, regardless of where it was caught or the time of the year. So it's quite prevalent, and in fact, much more prevalent than we were expecting. So then we had a look at seasonal variation in infected swordfish. And while the prevalence of infected swordfish is quite high overall, we do see that seasonal signal, right? Like higher prevalence in summer compared to winter. And that relationship is also reflected by the intensity of infection, which we measured as the number of spores within the tissue with the hemocytometer. So then we now want to model the risk or probability of harvesting an infected swordfish using generalized linear mixed models. So step one is we got the spatial catch locations of infected and non-infected swordfish that were provided directly from our industry partners under strict confidentiality agreements. We then paired that with corresponding environmental conditions for that day, and that came from satellites and regional ocean models. And then we used generalized linear mixed models to actually model the prevalence and intensity of infected swordfish. Now, I won't get into every predictor that was resulted from our final model, but this term here is by far the most interesting. So the best predictor here, it's the interaction between sea surface temperature and southwards current flow or meridional current flow. So remember, the East Australian current, that big warm ocean current, flows in a southwards direction. So what this graph is saying, if you look at that purple line, that's southwards flow. So if you're fishing in the East Australian current, southwards flow, and the sea surface temperature is high, you're almost guaranteed that most of your swordfish will be infected with this parasite, at least using our data set. And so now that we have our final model of infected swordfish, we then converted that into seasonal risk maps of the probability of harvesting an infected swordfish. So first thing I want you to note about these maps is the colour scale. It starts at 50%, where purple is less and light green is more. And it's quite obvious that that region of the East Australian current is a total risk hotspot, especially so in summer. So again, if you're fishing in the East Australian current in summer, you are very, very likely that most of your sword will have this parasite. So now that we know the environmental conditions associated with infected swordfish, we now want to see how these risk profiles might change under future ocean conditions. So now we'll move into our forecasts and climate projections. So first, I'll start with forecasts. A forecast is a prediction on a short timescale, usually ranging from days out to seasons in advance, so like a weather forecast. And the key thing about forecasts is that they're only concerned with environmental variability. Now, we used forecasts of sea surface temperature from what is called the Access S2 seasonal forecasting system. Basically, it's the forecasting model that the Australian Bureau of Meteorology uses to produce their weather and climate forecasts. So we got SST forecasts from them for the year of 2022 and ran our seasonal forecasting model, which looks like this. So again, the top row is the probability of infected swordfish. And again, you can see that general seasonal pattern with higher risk in summer compared to winter. And in this case, you do have relatively low uncertainty associated with those predictions as visualized by the standard deviation on the bottom row. So that's it for the forecast. Now I'm going to move on to the climate projections. So a projection is a prediction on a much longer timescale, right? From usually decades to centuries in advance. So think of a temperature projection out to 2100. And projections also include the effect that humans have on the environmental system through the use of greenhouse gas emissions. So we downloaded monthly fields of sea surface temperature and southwards current flow out to 2100 across a bunch of different Earth system models that are visualized there on the top. Now, each of these different climate models has an associated set of scenarios or SSPs, which essentially dictate, alternative futures for humanity based on greenhouse gas emissions and mitigation strategies that governments take. So I won't get into the nitty-gritty of the methods here for our climate projections, but we did, download the projections, we regridded them, remapped them, and bias corrected them before actually creating our projections ourselves. So now we'll look at the projected ocean conditions for this region of the fishery out to 2100 across a bunch of different climate scenarios. And I mean, as expected, this is for sea surface temperature anomaly. We do see that increase in sea surface temperature anomalies out to 2100 across all climate scenarios, with that expected acceleration of SSTs most prominent for the most extreme greenhouse gas emission scenario. So now I'll show you an example of the actual projections that we made. So this group of panels on the left is for the SSP 245 or business as usual climate scenario. So the column on the left, that's the probability of getting an infected swordfish. The middle column is the standard deviation or associated uncertainty. But the column I care about is the one on the right. That's the delta or the change in risk between projected conditions and current conditions. And I mean, again, it's quite clear that that region of the East Australian Current is projected to remain a total risk hotspot, especially so out to 2100. And so then we can rerun these models for the extreme climate scenario, SSP 585, and we see the same pattern, right? East Australian Current, hotspot of risk, but that risk increases out to 10% more in the long-term time period. And remember, prevalence is already predicted to be quite high anyway. So the fact that it's projected to increase by up to 10% more is, kind of worrying, right, for the fishery. So this brings me to the end of the swordfish section of my talk. And I guess the whole point of this project is that, it yields the first step required for being able to proactively adapt to the risk of catching a poor quality swordfish under changing ocean conditions. And the whole point here is to minimize wastage, maintain profitability of the fishery, and improve the sustainability of the industry under changing ocean conditions. Now, we only looked at one very small axis of environmental variability during my PhD, and there are many unresolved questions here related to, the host pathogen dynamics and how that interacts with the environment. So essentially, there's a lot of unresolved questions and things that need to be done to completely being able to resolve this jellymeat host pathogen problem. All right, so that's the end of the swordfish part. I'm now going to move on to red abalone. And this is what I'm working on for my postdoc at UC Davis. So if you're not familiar, red abalone, largest species in America, and they're an iconic species in California. They're a big deal, right? But due to a combination of, fisheries overexploitation and a range of environmental stresses like harmful algal blooms, seaside wasting disease, urchin blooms, marine heat waves, that has all led to the decimation of kelp forests, the primary food source for abalone. And so essentially their populations have tanked, right? And so that has led to the closure of the commercial red abalone fishery in 1997 and the closure of the recreational fishery in California in 2018. So all of this provides context for why our project exists on modelling and mapping climate refugia for red abalone in California. So we're funded by the California 5th Assessment on Climate Change, and the project is led by Michaela Provost, my boss at UC Davis, and we're working with a team from NOAA Monterey and San Diego. And so we're all about climate refugia, right? So refugia is essentially a place that remains buffered from the effects of climate change and can facilitate the persistence of a species under changing ocean conditions. For this project, we are using future projections of temperature, pH, and dissolved oxygen from this dynamically downscaled regional ocean model called UCSC ROMS, developed by people at NOAA. So it gives us, high resolution 3-kilometre projections of these variables on a daily basis out to 2100 across 3 different Earth system models. Now, when we create our maps of refugia, we're creating these maps for the entire state, but we're particularly interested in these 4 case study regions: Fort Bragg, San Francisco, Monterey, and the Channel Islands. And this is because, they have vastly different oceanographic conditions, different thermal regimes, so we can capture a broad range or latitudinal gradient of different environmental conditions. So first step for this work is we actually want to define what is refugia for abalone in the first place. There are many ways that we could have gone about this, and the approach that we have taken so far is a more mechanistic approach where we trawl the literature, we talk to abalone ecophysiology experts, and we find field and laboratory experiments that look at the physiological tolerances of abalone to, temperature, oxygen, and pH, and create a bunch of different definitions based on that literature and expert solicitation. So this is very much in progress. We've got 8 so far, But, these definitions, some of them relate to the pure survival of the species and are looking at marine heat waves. Others are more focused on fertilization success. Others are more focused on, promoting abalone with high growth rates. So, the point here is we want to create a vast array of definitions that capture a vast array of biological processes and life history stages. So today I'm going to go through one of these definitions, which relates to short-term variability in temperature. So our definition of refugia here is when abalone experience low short-term variability in temperature over 4-day moving windows, right? So not super variable conditions. And here we've got a 2-week snapshot of temperature in a yearly time series. So for abalone, the time period that we're talking about is based on what an abalone experiences today. So for example, February the 19th, and over the previous 3 days. Now, today's temperature matters more for the abalone. So we're going to weight this day more heavily, as indicated by the circle size. We then fit a linear regression to these temperatures, or the weighted temperatures, to then calculate the acclimatized or the expected temperature. So this temperature is what the abalone expects to experience, right? Based on the previous 3 days of temperatures. But you can see that it actually experienced a lower temperature than what it was expecting. So the difference between the acclimatized or expected and the current or observed temperature is what we're calling stress, as observed by the green arrow there. And so we can repeat this process for every day in our time series. We fit the linear regression, we get the expected temperature, get the difference between the experienced temperature, calculate stress. And so yeah, we do that for every day. And so we'll end up with a yearly or total time series of stress, right? The difference between the expected and experienced temperatures. What we can now do is calculate the integral, or the area under the curve in this entire time series, to create a spatially explicit map of stress for a year of interest, which looks like that on the right. So that's an example of stress experienced in San Francisco Bay area for 2050. And so then once we've created our stress map, we can convert that into a binary refugia map using a temperature threshold. So here we've said that refugia is when the abalone have experienced total stress below 10 degrees. And yeah, orange is no refugia, so the conditions are too stressful. Purple is refugia, so not as much stress. And yeah, we see this, delineation between the northern and southern part of San Francisco Bay around Point Reyes there. So we do that for every year out to 2100, and we can then calculate the persistence of refugia, right, on a range of different timescales. So here we've looked at historical, mid-century and end of century, and that's based on what CDFW are after. And you can again see that general pattern, right, where that delineation between northern and the southern part of the bay, with a loss of refugia in that northern part at the end of century. And this is also reflected in our delta maps, or the change in refugia of projected conditions compared to current conditions. And so red is essentially loss of refugia. So that's a example workflow of how we're going about this for one of our temperature definitions. I'll now show you a different definition that relates to fertilization success of red abalone. So here we've defined refugia as when abalone experience temperatures that are between 15.5 to 20.5 Celsius, regardless of the pH, or between that range there. And this again comes from the literature and expert solicitation, and it's particularly relating to like upwelling dynamics around the coast. So again, we can do a similar thing as I just showed. We create our yearly maps of refugia for each year out to 2100, and we can then look at a time series of refugia. So that black line shows the proportion of the year where refugia conditions are met across all of these grid cells. And so, out to 2040, conditions are generally conducive to promoting fertilization success, but we do see that steep decline with potentially, no more conditions conducive to fertilization by the end of the century. And then again, we can look at the persistence of refugia, where we've got high persistence now and in mid-century, but by the end of century, it's almost a total loss, which is reflected in our change or delta risk maps, at least for San Francisco Bay. So we're repeating this workflow for all of our different definitions that we've created, but what we eventually want to create is essentially ensemble all of these definitions together to create a mega map of refugia that integrates all of these definitions and all of these, biological processes together across multiple variables. And so this is an example of what that could look like. So on the left, we see ensembled projected refugia out to 2100 across our current list of 8 definitions and how that changes through time. And again, on the right, we've got persistence of refugia in our end of century time period. So that's where we're currently up to with this project. We still have a lot of work we want to do. We want to firstly experiment with different combinations of definitions. How does refugia change if we only look at definitions that are related to temperature, for example, or if we incorporate all definitions? Another thing we want to look at is kelp. That is, a big thing that is currently missing from our workflow. And, if there's no kelp, there's generally no abalone. So we're going to get historical kelp persistence from Kelp Watch and see how that interacts with our refugia projections. And lastly, we're also interested in the proximity of our projected refugia locations to historical, red abalone fishing ports. So, a lot of exciting work to come, essentially. And lastly, we are in the process of making an R-Shiny app for CDFW and for researchers and the public, that can host our maps and projections of refugia so people can go on at will and download, data they need and interact with that as they want to. So that brings me to the end of the red abalone section of my talk. Before I conclude, I did just want to give a shout out that my boss, Michaela, and I are running a workshop on how to work with Earth system models in R at Bodega Marine Lab in a couple weeks. So if anyone is interested in joining for Zoom, we are at capacity for in-person. Feel free to let me know. Otherwise, we will be making the notes, open source after the workshop. So yeah, thank you so much. Here are my contact details and keen to hear your questions. Thank you.
Wonderful. Thank you. I think we can dig in a little bit. So we're going to bounce back and forth between questions online and questions in the room. Roseanne, any questions online yet? Okay, so the folks online, if you have any questions for today's speaker, go ahead and put them into the chat. How about questions in the room? There we go.
Thanks for a really interesting and animated talk. That was really fun. Do you happen to know, have there been experiments on, say, certain thresholds for spore prevalence or spore density in billfish that actually produces the jelly meat? Can you be slightly infected and have no actual effects?
Yeah, I wish there was. Yeah, the short answer is no, but I have noticed in the past few years, there's a real uptick in research effort now on this whole Kudoa issue, at least in Australia, because, it's not just swordfish that get this parasite, mahi get it, kingfish. It seems to be the high value, bigger fish that get it. But unfortunately, yeah, no spore threshold information yet. Yeah, but I think that research is coming. Yeah.
All right, we have a question online first.
A nice talk. Does the ROMS model resolve the SF Bay outflow?
Sorry, can you repeat that? Sorry.
Yes. Does the ROMS model resolve the SF Bay outflow?
Yeah, that's a good point. I mean, yeah, the grid scale is 3 kilometers. I'll need to look, yeah, I'll need to look at that. Yeah.
Thank you.
Great talk. Going back to the kind of the parasite load and things like that. I guess what is like the vector of the parasitic infection? And I guess are the billfish their hosts? Like what do they go? Is this part of their life cycle to jellify like the meat, or is this just kind of like a side effect of some weird, just like over infection kind of thing?
Yeah. So the intermediate host is an annelid worm. And so the life cycle hasn't completely been resolved, but the current, going thing is that the spores, float in the water column, they get into an annelid worm, they then develop into an adult spore that gets released. Somehow they get into contact with the swordfish. We think it's due to pheromones, but that hasn't been resolved. But yeah, it is part of their life cycle to jellify the tissue because there are other species of Kudoa that also cause jelly meat in other fish. So yeah, we think, they jellify the tissue, the spores can then get out of the fish and then go infect a worm elsewhere. That's like the going idea. Yeah.
Hang on.
So the infection, it doesn't necessarily kill the fish. It's just when the fish dies of some other kind of
Exactly,
reason.
yeah. There seems to be a negligible effect on the fish when it's alive. There can be, I've seen fish that are completely riddled with this parasite and, they seem to be good, healthy fish, but then you wait a couple days after they've been killed and they just turn to mush. So, yeah.
Thanks. So since you said about 70% of the fish you sampled were infected,
Yeah.
they can't all have jelly meat, right? Or the
Yes.
fishery would just say, this isn't worth it. We're not doing it. Are you like, I'm curious about the relationship with your fishing industry. Are they interested? Are you distributing the results of your work to them? Are they asking for more? They want to use some of these maps and projections to adjust their practices?
Yeah, for sure. I'm so glad you brought that up. So this whole PhD project came to be from a conversation between my supervisor and one of the fishers who were like, oh, we're seeing all this jelly meat. We don't know why. Like, we want to figure out what's causing it so we can stop it from happening. So yeah, like throughout the project, we had an industry representative. And so, each year we would provide updates to the fishers, and every time I would sample a couple times a month, sharing results and stuff with them there. But yeah, they are definitely interested in like the forecasts and projections, but they're never going to change where they're going to fish, because in this case, it's a multi-species fishery. So they'll just, if they're getting heaps of jellied swordfish, they'll just switch to yellowfin tuna or something else like that. But, one thing that they have changed, which is cool, is that they process the swordfish much quicker now. Like before they used to catch a swordfish and they'd kind of leave it in the sun for a bit and decapitate it a bit. But now they just process it straight away as soon as they catch it, get it iced, So they have changed their onboard processing because of this, which is cool.
I want to jump in there real quick and then I'll hand it off. So post-mortem handling of the fish. So we're seeing some benefits of quickly cooling the fish meat to be able.
So. Yeah, for sure.
I'm curious if you have looked into any of the research or stuff from the Pacific hake fishery.
Oh, no.
So it is also a Kudoa parasite, and Pacific hake out here is notorious for jellifying, like pretty immediately. So when you're on survey, that happens. And so hake is not a high in meat though, right? So like that is easy to get processed really, really quickly out at sea, but that's the only way that we keep the hake fishery from like not jellifying. So I was just curious if you guys have looked into that.
Yeah.
Because if you can't get rid of it, processing fast might be the only thing that you can do.
That's it. Yeah, that's a really good, interesting. I had one conversation with one of the processors and I was like, oh, could you just snap freeze the swordfish instead? And they said no, because they want to export the swordfish fresh. Like the whole point of sashimi is that it's not frozen. So they were pretty hesitant for that. I guess, yeah, I do know that the jellied swordfish that they do get before they send it to the restaurants get sold off as pet food. So they're still at least making some money from it, but yeah, just at a reduced price. Yeah, interesting. I wasn't aware of that.
Other questions in the room? Roseanne, any other questions online? All right, I'm gonna wiggle down.
Thank you for the talk. Very nice. About the abalone, they're going to be adapting over time to changes in heat there. So your modeling is going to be based on a changing endpoint over time. How do you deal with that?
Yeah, I mean, currently we're using a fixed baseline. So yeah, honestly, we just need to experiment with different thresholds, I think, and just see how that influences the projection. So yeah, essentially a lot more to do. Yeah.
All right, let's look at another question online. Go ahead, Roseanne.
Abalone live a long time, so they don't need to spawn every year. Could there be temporal variability in refuge status? So some places are okay some years, but not others.
Yeah, that's a really good point. I mean, I guess I haven't really thought of it that way, but yeah, thanks for raising that. Yeah.
All right, any other questions in the room? And Roseanne, no more questions online? All right. Can you go back to your contact information for us and just put that up? Hang on, let me show you a trick. There we go. All right, everybody. Thank you so much for being here. If you have questions and follow-ups, contact information is now on the screen. But thank you so much for being here. Lots of things for next week as well. Hope to see you again. For folks online, thanks for being here. Thanks, everyone. How about one more round of applause? All right, we'll see you next week.